currently serve as Chief Scientific Officer and Head of Translational Sciences at Immunologix Laboratories. We focus on large molecule bioanalysis, PK, immunogenicity, and biomarkers. And our translational sciences team comes with decades of drug development experience, and so we also serve as scientific consultants to our clients. I would say is a case of ever-increasing complexity. So if you think 25 years ago, we were in a place where we had recombinant proteins, monoclonal antibodies, systemically administered, not necessarily fully humanized, a very different place from where we are now, where those modalities have expanded to include cell and gene therapies, nucleic acid-based therapeutics, complex molecules, bispecific antibodies, trispecific antibodies, antibody-drug conjugates, antibody oligoconjugates. Peptide drugs have expanded to all kinds of shapes as well. We have circular peptides, and all of these now need to be understood in a way that we thought we understood things 25 years ago. And so while technologies have also grown along with these modalities, we've had to fold in flow cytometry, nucleic acid measurements, real-time PCR, all kinds of approaches that we didn't need 25 years ago. But what I think has been most interesting is that we've been trying to do this in the context of guidance documents that were written only for protein therapeutics. And so where we are now is a place where we need to determine the best way to understand drug exposure, development of immunogenicity, biomarkers that can be decision-enabling for a drug program, but sometimes leaning on paradigms that were not built for the purposes that we have now. And so that is where we spend a lot of our thinking time and partnership time helping to navigate through those complex places. The growing complexity means that we need to work from first principles all the time. The existing guidances, as I mentioned, were developed for simpler protein therapeutics. It was a place where you could set some standard rules and everybody could follow that kind of template as they developed their drug. And it seemed a path that could be understood and replicated with the next drug. All of this is off the table now with these new modalities. These guidances and approaches were developed without even imagining the complexity that we have now. So at this point, it's really a case of understanding what is my drug, what is its target, how are we administering it, where are we administering it, what is the relative dose level, what is it that I need to understand so that I can understand impact on patient exposure, safety, efficacy. All of these aspects need to be answered in a different way with these different modalities. And a lot of the traditional ways that we've thought about things do not apply. Historically, you would dose a monoclonal antibody generally at high levels, and you would be able to measure it relatively easily, and you would expect there to be an immune response. You knew how to go about measuring that. Now we have some modalities that are truly microdosed. The challenge of even being able to prove that you have dosed your drug at the level that you want, much less than having a biomarker that proves it got where you needed it to go, it had the biological effect that you expected it to have, and then if there is any immunogenicity generated against it, what is the correct level of relevant immunogenicity? Do we have a technology that could measure that? And all of these pieces really need to be thought about in the context of the whole program, how all these data sets can be generated, how they speak to each other, and how together they tell the whole story of what's going on. And there is no rule book for that. In thinking about how the modalities have become so complex and all the different disease indications that we're targeting. It's really been a fantastic journey as biology knowledge has grown. We're able to develop much more targeted therapies. We have genetically defined diseases that we can really address directly. But again, as I said, we don't have guidance that can address all of these situations. And so that is where, as scientists, we need to go back to the basics and understand what is the question being asked? What does success look like for this molecule at different stages? In early stages, you want to understand, did we get the exposure we expected? Did we see any safety events? But very early on in drug development now, we want to get the first hint. Is this actually having an effect that we need it to have so that we can kill bad drugs early and progress good drugs quickly? And so that's where biomarkers come into play, because they can give us an understanding of, are we hitting the biology that we intend, which then de-risks the spend that we put on that program to move forward to very expensive, larger clinical trials. This is going to look completely different for every drug type, every pathway that it's interrogating, every patient population. And you have to thoroughly go through all of those aspects. I often think about it as though I were doing a PhD thesis again. I need to define all the variables of my project. I need to think deeply about the questions I need to answer and all the different ways that I could go about interrogating that problem and getting to the right answer. And I think the challenge that we have had is that that is frightening to a lot of people in the regulated space, because there's always this concern that, how will I satisfy the regulators? And if I have to do something unique for my molecule, how am I going to bring the regulators on that journey with me? And so this comes to that fit for purpose concept too, is how do I demonstrate that what I'm doing is in fact fit for my purpose? And this is where I see a lot of industry trying to over-apply existing rules and guidance and put square pegs in round holes. But when you do that, you really risk not getting the correct answer. And so I have always been a great advocate for engaging with those regulators very early. The regulators say it all the time. They say, please come talk to us early. And I have found that if you do that, and you are scientifically sound, and you set out a strong strategy, you have it technically supported with your technology platforms and the way that you've developed and validated your assays, you can take them on that journey with you. And I think that's inescapable now. Long gone are the times that we can tick certain boxes and know that that's going to be okay. It really is beholden upon us to think it through and be confident in our science. So the challenges for a drug developer to bring a molecule from the research bench to the clinic are obviously too numerous to articulate here. But if we think about how a CRO partner can help in that space, there are a few key areas that we participate on the regular basis. The first thing is a lot of the experiments that can be executed on a research bench and answer a question quite elegantly cannot translate to the context of a clinical trial, much less a global clinical trial. You can't do things in real time. You need to understand, how could those samples be collected? How can they be stored? How can they be shipped? Can they get shipped in a certain amount of time under the right conditions to arrive intact at the testing laboratory? And so that's a space where our translational team can be really helpful, because they have executed worldwide clinical trials themselves in their drug development careers and have really vast knowledge on what is implementable in the context of a clinical trial. And so with a lot of our early stage clients, that can be a lot of the conversation. They may have a great deal of really good ideas that interrogate the biology, but we help them understand how and what can they actually execute in the context of a clinical trial. The other place that I think where we can help is in understanding, how do we support our assays and their life cycle over time? So you imagine a typical drug development program is going to span 10 years or more. And so as they're developing their methods they're going to want to use through the entirety of their drug development program, they need to generate critical reagents. How do you generate those reagents? What do they look like? How do you sustain them? How do you have a stability program that ensures that those reagents can be used over the life cycle of the program? In addition, depending upon patient population changes, evolving regulatory expectations, your assay may need to be iterated over time to keep up with those new expectations. And so that's where a CRO who lives and breathes multiple programs at all stages of development all the time really has that kind of experience and knowledge at their fingertips to be able to help a client navigate through a space that may be new for them. The thing that I love most about working at Immunologix is that we have had the luxury of reinventing what it is to be a CRO partner in the large molecule space. And the thing that makes us different is that we do have our integrated translational sciences team along with our laboratory experts. And the way that that works is that these translational scientists have all come from decades of drug development experience sitting on the sponsor side. They have walked in the sponsor's shoes. They have carried molecules from late research all the way through to post-market. And so every regulatory interaction, every sort of internal angst that a drug development company may experience, we have experienced. And we understand the pressures that are on our sponsors, not only from the scientific perspective, but the internal pressures they may have on their portfolio, that the leadership may be bringing, that their investors may be bringing. And it really is the integration of the science with the pragmatic realities of drug development that we can provide that differentiates us from others, because we've been there and we've done that. And we can really help our clients navigate a place that is scientifically sound while accommodating some of those other complexities that can come into play. The way that we then bring that together with our internal scientific team that is actually developing and executing the assays in the laboratory is that every program that we bring into our lab to develop a new method, we have a very extensive intake process. We put this client through a multi-slide list of questions where we need to learn about, tell us about your molecule, tell us about your mechanism of action. What is your intended patient population? Oftentimes they'll be starting in healthy volunteers, but they know what populations they want to go into. The sooner we know that, the better we can develop an assay that's going to serve them for both their early and their late needs. In addition, we want to understand what biomarkers they're looking for, all kinds of aspects of the molecule so that we can ensure that we are developing the assays to really address the questions that they need to answer and make very confident decisions based on the data that we generate. We then bring all of that information in-house. We have an internal kickoff meeting with what we call our triad of scientists. So our triad of scientists includes that translational scientist who helped bring all that information in, along with our lead development scientist who will lead all of the bench work, and then our principal investigator, who is the scientist who will be that single point of contact for the client, bringing them all the updates of the data, walking them through progress on the project, talking to them and understanding their evolving needs in real time. And it's with that project team of scientists that we execute our programs. In addition, a very unique offering that we also give to our clients is that our senior scientific team that includes all of our translational scientists as well as our scientific leaders in operations hold daily office hours at our organization. So reminiscent of academia, where you would go to your professor to ask for help, we hold these office hours every day to enable our internal scientific staff to have access to all the expertise under our roof on the daily basis. So if they're running into a challenge or have a question, they can come in real time and pick the brains of 150 plus years of experience on any given day. In addition, we also invite our clients to come to office hours. So if it's a client, they may have an ongoing laboratory project that they want to come talk to the broader team about, but we actually open it up to anything that they want to talk about at all. So if you are a client of Immunologix, you can use this consulting team to get input on anything. So we've had folks come who want to talk about, we have a new program coming down the pipeline. Here's some thoughts we're having on our biomarker plan. And very early on, they can get a whole panel of experts telling them, what they think would work or not, both based on science as well as the logistics of executing it. And so that really builds a level of partnership that is, I think, unique in the industry. But in addition to that, it's such a synergistic kind of experience because the client is benefiting from a breadth and depth of experience they really can't access in any other way. But our own scientific team is benefiting because we're getting firsthand interactions with the real problems that drug developers are facing every day. And so I think that's what makes us a little bit different. One of the things that as we've evolved as an organization that we're really committed to is in that educational realm and providing forums for scientific exchange. Internally, that's internally we have, beyond the office hours, we have developed a comprehensive scientific training program that allows our employees in real time every week to be getting new information, vignettes from our own projects, as well as foundational training so that they have that to carry forward. We are truly committed to scientific education. I think as will resonate with some of the other things we were discussing, so much of doing modern day bioanalysis really does require continuous learning. So we have several internal programs that we offer to our own scientists so they can learn constantly and upskill on all fronts. But in addition, we do a lot of external facing education. So we do hold virtual seminars on a fairly regular basis. We call it the Rethinking Bioanalysis series, where we openly address the changing landscape and question the way things have always been done, and is it time to think about doing things in a new way? And then for what will now be our third year and moving forward, we began the Immunologix Symposium. And so that is held once a year in the Boston, Cambridge area. And that is an open forum. It's free to attend. And we put together a scientific program that really is meant to address key questions that are front and foremost in drug developers' minds. The way that we choose the topic is really based on our experience over the prior year of the questions we're being asked the most, the things that we feel people are desperately seeking more guidance on. And then we have invited speakers from across industry as well as former regulators who come and talk about where they think things are going. We provide a lot of time for open discussion so that it's really a very collegial, fun sort of event for people to talk about what's front and foremost on their minds in an environment that is much more open and fluid than may happen at a typical sort of large meeting that you may go to. And in addition, we host it for free. And we provide food and beverage for the day. And so it really is almost like a bit of a scientific retreat that we provide. I think we all are aware that a big factor that comes into play is often cost. This is a reality in the industry. I would say, though, that what is most important is that scientific partnership and ways to understand, is this going to be a good partner for me, that I think about is when you're in that first interaction with that potential partner, how many questions are they asking you? Are they trying to understand your program? Or is the partner doing more talking than the potential client? I was on the drug development side for most of my career before coming to Immunologix, and it becomes very clear when a partner is kind of trying to fit your program into their processes and their approaches, because it means they can move faster. And I think the partner you're looking for is the one that you can tell is really committed to understanding what you need, how they can help you. And in addition to that, beyond the science, they're also trying to understand, what's your working style? How do you like to communicate? What kind of cadence of meetings do you want to have? How do you want data delivered to you in a real time? And all of these components that really speak to developing a relationship on multiple levels is the key factor in selecting a partner. While many of us will find that sometimes we do get steered to a partner that isn't our first choice because there may be other factors at play within an organization, I can say that we have many examples of clients who, for various reasons, went elsewhere, and then they show up on our doorstep again because things maybe got complex or they didn't go as well as they had hoped. They're coming looking for thought partnership. They're looking for that partner who's not just there to knock out an assay for them, but who's there to really think through the complexities of their program and try to find them the best solution to get them to that next critical decision.